The core of the innovation is the use of special enzymes, which were discovered by researchers at the University of British Columbia, UBC, and collaborators. The problem: the body's immune system identifies an organ's blood type by the antigen sugar molecules that coat the blood vessel walls. If the donor organ's antigens don't match the recipient's blood type, the immune system will launch an attack, leading to rejection. Type O is the universal donor because its blood cells and organs lack these RB antigens. The solution: the researchers used a technique called enzyme-converted OECO. They perfused the type O donor kidney with fluid containing highly efficient enzymes. These enzymes act like molecular scissors, effectively stripping away the antigens from the kidney's blood vessels. The result: removing the antigens essentially converts the organ to a universal type O kidney, which could then theoretically be transplanted into a recipient of any blood type of B, of, or without an immediate, severe immune reaction. The first-in-human experiment. Researchers conducted a first-in-human experiment to test the modified kidney. The test: an enzyme-converted type O to type O kidney was transplanted into a brain-dead recipient with a blood type mismatch, a type O kidney into a type O recipient. The initial success: the ECO kidney was accepted by the body and functioned well for 2 days with no signs of rejection, demonstrating the concept worked initially. This was the first time this process had been successfully demonstrated in a human model. The challenge: on the 3rd day, the antigens began to reappear on the organ, which triggered an immune response. However, the rejection response was less severe than a typical blood type mismatch, suggesting the body was trying to tolerate the organ. Why this is a major breakthrough? This research, published in the journal Nature Biomedical Engineering, is considered potentially game-changing because 1. It expands the donor pool. It could unlock the use of organs that are currently discarded due to blood type incompatibility. 2. Reduces wait times. By removing the blood type barrier, it could significantly reduce the long wait times for kidney transplants, especially for type O patients who make up the largest group on wait lists but can only receive type O organs. 3. Addresses inequity. It could particularly help minority ethnic groups who often have rarer blood types and find it harder to get a compatible match. Next steps. While the initial results are promising, there are still challenges to overcome before this technology can be used in a full clinical setting. The researchers need to find a way to make the type O conversion stable for the long term to prevent the antigens from reappearing and causing rejection.